We're going to move on to Xiang Deng, who's going to talk about ALS5 and mouse models, some molecular insights into neurodegenerative disease. Very briefly, Dr. Deng is a professor, a research professor of neurology here at Northwestern. He's really made seminal observations about the genetic underpinnings of ALS, and look forward to hearing of his most recent work, his new work. So Xiang, take it away.
Okay, today I'm going to talk about something we did on ALS5 and its mouse models. And I want to show what we have learned from this mouse model and what implications it has in understanding the mechanism of ALS5 and potentially the implications in therapeutic development. And first, ALS5 is the most common autosomal recessive ALS, and it's early onset. The disease onset is about 10 years old, and can be earlier, like 3 to 4 years. And the progression is very slow. So the patient with ALS5 may wheelchair bound by like 20 to 30 years old. And the duration of the disease is very long. They can be 30, 20, 40 years old, 20 years after disease onset. And the disease is upper motor neuron predominant. And this gene has been studied, and the gene is mapped by Dr. Siddique's group in 1998. The gene is mapped to chromosome 15. And 1 year later, and there's a disease called spastic paraplegia type 11. And this spastic paraplegia is a complicated SPG, and with a thin corpus callosum and with a mental impairment. The IQ mostly is around 60. And they also mapped this gene to chromosome 15. Look, so we can see there's an overlap here. And in 2007, the Stevani group, they identified the mutation SPG11. And we can see most of the patients, they initially show spastic paraplegia. And normally after 10 to 15 years after disease onset, they may show the lower upper motor neuron signs. So actually this one, ALS5 and SPG11 is the allelic disease, and meaning it looks like a different disease, but it's caused by the same gene mutation. And in addition to motor neuron involvement, and they may also have the sensory involvement. So some families, they characterize as Charcot-Marie-Tooth disease type 2. And in some families or patients, they show juvenile Parkinsonism. And interestingly, we can see this patient with SPG11 linked mutation, their symptoms are dopamine responsive. And interestingly, if you do the deep brain stimulation, they have excellent effect, and this effect is sustainable. So when Stevani, they identified this mutation, and because we can see the SPG11 and the ALS5 is the same, looks like it's overlapping locus, and clinically they are overlapping. So we also tested if ALS5 is the same mutation, same gene mutation in the family. So we tested all 3 families, and we found indeed, we found all the families with initially used to map ALS5 has mutations, homozygous mutation in SPG11 gene. And we can see this is the autopsy sample from ALS5 patient. And we can see the cortical spinal tract degeneration. And interestingly, we can also see the dorsal column degeneration. That will explain why some patients, they may show Charcot-Marie-Tooth disease. That means the sensory column, sensory axons are also involved. And this is a spinal cord from one ALS patient. We can see the lower motor neurons are largely lost. Here we can see one lower motor neuron. And so that means even though they clinically they show like different phenotype or overlapping phenotype, actually the lower motor neuron is indeed involved. So diagnosis, if we say diagnosis at the later stage, ALS5 is quite reasonable. And in addition to motor and sensory system, we can also see the white matter. This is a control. The white matter are greatly diminished in the patients. This is ALS5, this is a control. That's the white matter here, and here is much more reduced. And this is the corpus callosum, and the corpus callosum normally is like this thick, and here is almost gone, very, very, very thin. And here is the other system. This is lateral geniculate nucleus. This is a control. We can see this big. This is very, very, very small. And the microscope, you can see the nucleus, the neurons in this nucleus. This is control present. These are largely lost. And this is substantial diagram from the patient, the ALS5 patient. You can see a spatial structure. This is actually the E staining. We can see the structure we have never seen before. And this looks like a new structure. And so we characterize this structure. And this is actually staining showing the structure like this. Obviously, it is not. Cystatin C positive is not vanilla body. It's not over-localized with the TDP43 in the nucleus. This is the structure. But it is positive with ubiquitin and P62. So we tested a large number of antibodies to identify what this structure is. Eventually, we found, looks like this cell is a chromogranulin A positive, and suggests that this structure may be large densicle vesicles, and the ubiquitin and chromogranulin A identify this structure. And we did a cell biology study and tested a large number of almost all the subcellular organelles. We can say this is a lysosome. It's not lysosome structure. This is a protein spastin labeled with M cherry, and we can see if they are colocalized. Obviously, it's not, and it's not colocalized with the ER, not colocalized with Golgi, not colocalized with the synaptic vesicle, not colocalized with the endosome and the autophagosome. They're not colocalized. But when we test it using the large densicle vesicle markers, you can see this CHGA is nicely colocalized. And in addition to CHGA, the secretogranin 2 is also a large densicle vesicle marker, is positive, almost 100%. And this is also a large densicle vesicle marker, the SLP4, very nicely colocalized. And this is in the neural 2A cells. And when we cut the primary neurons from the mice and transfected cells, we can see it's nicely colocalized. So it is transported to the dendrites or axons, neuritic process. So meaning it is really most likely this is the structure is the patient that we observed is large densicle vesicle aggregation. And in the patient, we can see pathology. Yes, it is not consistent with the cell biology study. This is the pathology, and this is synaptic vesicle. They're not colocalized, not with a fuse. This is structure. This is a fuse, not with the autophagosome. This is structure. This autophagosome. It's not lysosome also. We can see this is cathepsin D. It's a cargo in the lysosome, and it's not obviously selective. And in addition to CHGA, we can also see the substance P is a cargo in the large densicle vesicle markers. They are positive for this structure. And the large densicle, we can see this is large densicle vesicle, synaptic vesicle, and large densicle vesicle markers is much more larger. Normally the synaptic vesicle is about 40 nano, and this one is much larger. It's about 100 nanometer, and it contains the neuropeptide or neurotrophin factors, and will play an important role with the neuronal function and modulate neuronal activity. And then previously, a large number of studies using the GWAS or association study, such as some of the cargoes, like the neurotrophin some polymorphism may be related with the neuropsychological disease. But there's no direct evidence showing any mutation associated with the density vesicle marker are really linked to human disease. And if SPG11 spartaxin is true, it means this one is the first molecule that link the large density vesicles to neurodegenerative disease. So because the human samples are limited, and also there's some quality problem for test additional markers for this for validate the structure. So we developed the mouse model, and using CRISPR-Cas9, for example, this is wild type. This is one base pair insertion, 4 base pair insertion, 8 base pair deletion, and 17 base pair deletion. So these are we kept the mice with the frame shift mutations, and we can see this mice develop disease. It's really, and it hunches back, and the eye infection of the eye. That means they cannot groom the eyes, and also the movement problem by like 1.5 years. And like in the ALS mice, we know SOD1 problem is the best one. And the other mouse model, we made so many, and most of them they do not develop typical disease sign, or the signs may not be directly related with the disease. And this one we just simply knock it out, like what we occurred in humans, and the mouse develop disease. So this is a rare replication of a human disease, ALS disease, in the mouse model. We can say this is the curve showing the mouse develop disease, the loss of the disturbance of the balance or whatever, and it can be like 400 to 500 days, and the mice will uniformly die by 20 months plus minus 2 months. And maybe like SOD1, it's very consistent. And then we check the pathology in the mice. What we can see is the C2GA, this is the motor cortex. We can see almost exactly the same as what we saw in the human autopsy samples. And this is middle brain, C2GA. This is spinal cord, those anterior column. This looks like a big motor neuron maybe. And this is a dorsal column, spinal cord dorsal column. And they are all C2GA positive. And they're also secretogranulin 2 positive, motor cortex, and the motor cortex, and also granulin positive. So these are all not densical vesicle markers. So this one, obviously, we are pretty confident this pathology is the aggregation of large densical vesicle markers. And we also use confocal study secretogranulin and P62 granulin P62 are nicely localized. And this is a different like motor cortex, spinal cord, cerebellum, like Purkinje cells, and hippocampus neuron. The neurons in the hippocampus, they're all almost identical. You can see these are neurons, big neurons. They're all identical. And it's not these vesicles, as we saw in the cell biology, is not the lysosome. It's the independent of lysosome, because the lysosome is labeled with the red. This is cystatin C, cathepsin D, and it's red lysosome. And this structure is the pathological structure, obviously they're independent. And in the EM, in the transmission EM, we found like this structure with a large number of vesicles, but looks like not exclusively the EM only, but there's some other structure like mitochondria. Looks like a mitochondrial structure, but they are very uniform. They're quite pretty uniform with the large densical vesicles. And the mice, we can also see the activation of astrocytes. We can see this is the different layer, and this is the control different layer. You can see the increased, the astrocytes are increased or activated, especially in the axonal part, like the corpus callosum here. And also the microglia is also activated in the especially in the white matter area. You can see this is the cerebellum, and this can be clearly seen, and we can see the structure here, and compared to here, they're all different. Obviously they're obvious. And so what is the consequence of this genetic mutation and loss of spartaxin in the humans and in the mice? So, for example, this is a neuron. This is a Purkinje cells, because these cells are well separated from others, and there's no overlapping, so it can be easily identified and can be analyzed. We can see this is the Purkinje cell, this big, and so many branches of the dendrites. And here is the neural Purkinje cell from the knockout ALS5 or SPG11 mouse model. You can see the branches are much less, and much less. And we can see if we take this area, we analyze, what we can see is the spine, like these dots, spines are reduced. You can see the spine density is reduced, the width is also reduced, and the length is also reduced. So most likely, the loss of this protein will lead to the synaptic functional impairment, because the structure is changed, the spine is reduced. And this is not only only in the Purkinje cells. Actually, the whole brain, they're almost identical, but because the other area we cannot definitely define the neuronal cell type, but obviously they're reduced. For example, here is a hippocampal neuron, and this is there's so many branches and so many spines, but here they're much less. And what is the consequence of the loss of this the protein and the pathological structure in the mouse brain? So we collaborated with Dr. Minqing Jian and C.J. Heckman in the physiology department, and they measured the mouse spinal cord, the anterior root, the ventral root, and this is using the sacral segment. This is a sacral segment 2 and 3, and both sides. And this is the control. What they observed is these mice, even though they're not, it's not moving, but they have synchronized firing from the anterior root. You can see the firing synchronized. If this one is firing, the other one is also firing, synchronized. But this is not occur. This did not occur in the controls, and because why the mutant mice had synchronized firing, the obvious possibility is to increase the sensitivity of the lower motor neuron to the stimulus, so the stimuli in the environment or in the milieu of the neurons. And one obvious substance is the substance P, because substance P is a excitatory neuropeptide, and when this density vesicle is aggregated, they may affect the transport of density vesicles and the release of these density vesicles. And substance P seems the only neuropeptide which is only 11 amino acids, so can penetrate into the spinal cord, and then we can analyze. So we tried to analyze this one. So we use this is the wild type mice, and this is knockout mice. And when you put substance P to excitate the lower motor neurons, we can measure the activity. And yes, indeed, we can see this one. Yes, when you put substance P, it can excitate the lower motor neuron activity, but in the SPG11 mice, it's much higher. So suggesting the post-synaptic the the receptor is sensitized, and the receptor is urocinin 1 receptor for substance P, and we can see the firing is much more increased, and the firing time is much longer. So this has a very important implication, and because the mechanism suggests the sensitization of the postsynaptic receptors, and here we showed that at least the substance P receptor is sensitized. If this is the mechanism underlying the disease, and this may have important therapeutic implication. The first, we know if this was the urocinin receptor 1 substance P receptor is sensitized, it can well explain some of the upper-motor neuron signs. For example, spasticity. Why there's no stimulation, you just rest there, the patient is showing spasticity. That means the postsynaptic receptor that sensitized, the sensitivity is increased. And that's also will explain the hyperreflexia in the SPG11 and ALS patient. And of course, it can also partially explain some of the weakness because of the problem. And therapeutically, we can use theoretically, we can use the urocinin 1 receptor antagonist and to block or partially block this sensitization of the urocinin 1 receptor. And notably, there's a one drug called aprepitant, and this one has been approved by FDA for anti-emetic and anti-exalytic drug, and is now currently in use, approved for many years. And so this one suggests the pathway involvement and the drug development may go through this system. So overall, I showed some data from the human study, and we found the genetic mutation, and we found the dense-vascular involvement potentially in the ALS patient. We made the mouse model, and we validate the dense-vascular problem in the mice. And this spastin is the first molecule that links dense-vascular marker, dense-vascular to disease. And through the mouse model study, we found the postsynaptic receptor activation and the sensitization, the typical example like a substance P pathway activation. And this one suggests the antagonist for treatment of this disease. Normally, we want to test in cell or animal model again to see that the efficacy and the safety, because this one, aprepitant has already been tested and a good safety profile in the patient. So this drug can maybe directly tested in ALS patient for improve or ameliorate the symptoms in ALS patient. And I guess I'll stop here, and I would like to thank Dr. Thibaut Siddiq and Naila Siddiq that they initiated this genetic study and the pathological study, and Dr. Eileen Bijo did the pathological study, and Yongxi, Guoshan, Hong, Yi, and Yan, they did the genetic study and the mouse model study, and Dr. Yunqiao Ma and Dr. Miller, they did the cell biological study. Dr. Minqing Jian and Dr. Xie Heckman did the physiological study. This study is supported by ANH and the External Foundation. And thank you. I think I control the time.
Thank you, HX. That was wonderful. Any evidence for these abnormalities that you've uncovered in the in ALS5 in any of the other mouse models of ALS?
No, this one seems is a unique structure so far we found in the ALS5, but there's something maybe similar that is there's maybe also in SPG, so-called SPG15 and SPG48. They may have some similarities, but that one needs to be validated in the mouse model.
Okay, great.